Hi everyone, welcome to my channel Pure Biology, and today I come with the continuing video that is of conjugation. And here I will try to explain about the another two processes related to the conjugation. Obviously here the DNA transfer occurs between the HFR and F minus conjugation, and another process also the transfer of DNA from F dash and F minus. Okay, F dash and F minus bacteria. So here today's topic is the conjugation, how the DNA transfer from the HFR to the F minus conjugation. So before going to this conjugation process, first we have to understand what is the HFR. Okay, HFR means that the high frequency recombination. Okay, or you can say that HFR is the presence of high frequency recombinant DNA that present within the bacteria. Generally what are called the F plus. F plus bacteria, that they have chromosomal bacteria, also present F plasmid bacteria or the F factor. So this is the F plus bacteria. But HFR means here the plasmid and the chromosomal DNA, they already recombinant, and recombination by the process of recombination they form the recombinant DNA within the bacteria. Okay, so this type of bacteria generally known as the HFR bacteria or the high efficiency bacteria. So here the process occurring between the F minus plasmid and the HFR bacteria and the strain. So here this is the rough sketch. First you just you just see what is happening when the F plus that is converted to HFR. So this is the plasmid or the F factor, and this is the chromosomal DNA. Okay, so by the process of recombination, the already HFR or the high frequency recombinant DNA is formed, and this situation it is known as the HFR bacteria or high efficiency bacteria. And when the conjugation process is occurring between the HFR and F minus, so this recombinant DNA that is transferred within the F minus bacteria. Okay, and in that situation the F minus maybe remain in the F minus bacterial form or maybe form into HFR bacterial form. So it is happening how, if the chromosomal DNA or the bacterial chromosomal DNA only transferred within the F minus, then the F minus bacterium is only formed. But if with this chromosomal DNA, the F plasmid that is also transferred within the F minus, then HFR bacterium may be formed. Okay. Now it is saying what in the paragraph, we just want to see what is saying, that the high efficiency strains transfers of single stranded DNA from an HFR donor to the recipient and begins from the origin of the transfer within the F plasmid and proceeds. So here the transfer of single stranded DNA is seen just like the F plus and F minus conjugation, and this begins the space. This is called the origin of the transfer, and the origin of the transfer within the F plasmid and proceeds. So here the origin of the transfer I explained in my previous video. You just check out which one is the origin of the transfer, that is from where the nicking of the strand is occurring and the transferring of the DNA start. This is called the initiation area, or the, you can say that, origin of the transfer region. And now in matings between the F plus and F minus bacteria, only the F plasmid is transferred to the recipient. In case of, so what happening in F plus and F minus bacterial conjugation? They are only F factor of F or F plasmid that can be transferred from F plus to F minus. Okay, but in here the F minus, in the F minus strains and HFR, both the within the both strains, What is happening? Part of the F plasmid is transferred last after the entire bacterial chromosome has been transferred. Okay, if bacterial chromosome is completely transferred, then the plasmid transferred with this bacterial chromosome. And because only a part of the F plasmid is transferred at the start, the F minus does not become F plus unless the whole chromosome is transferred. So what is saying that? Just remind, if only one part of this, I just here mentioned the F plasmid by this red color. Okay, so what is happening here? If this F plasmid is completely transferred from the donor to the recipient, okay, then this F minus may be converted into the F plus. But this is possible only after entering this chromosomal DNA into the recipient from the donor. Okay, because in the first case only few or some of the plasmid that can enter from donor to the recipient, not the whole. And after completion of the transferring the chromosomal DNA, then the remaining part of the F plasmid, they may be enter within the recipient. Okay, but complete transfer takes about 100 minutes and the conjugation usually breaks before the process is finished. Okay, so what is thing that the conjugation bridge, okay, this cytoplasmic bridge that not remain up to the total transfer of the DNA from donor to the recipient. It not remain stable. It is breakable and it not remain stable for the complete transfer of the DNA. And so that's why a complete F minus plasmid usually is not transferred and the recipient remains F minus after the replicated donor chromosome enters the recipient. So what is saying that so after entering this chromosomal DNA from the donor to the recipient, they may remain in F minus formation, or if the total completion is possible, they may be formed into HFR form. Okay, so this is the conjugation process that is occurring between the F minus and HFR bacteria. Okay, so the next part is that I will try to explain about the conjugation process between the F dash and F minus. Okay, F dash and F minus. Okay, so next process is that F dash and F minus conjugation. So here the F dash and F minus conjugation is identical, just like F plus and F minus, and bacterial genes on the F dash plasmid that are transferred with it and need not to be incorporated into the recipient chromosome to be expressed, and the recipient becomes F dash and is a partially diploid merozygote since it has two sets of genes. Okay, so what is saying that F dash means it carrying the two gene but in the recombinant way, or the by the process of recombination. Here the chromosomal DNA and the plasmid DNA, they are all bind together and form the recombinant gene or DNA, and F minus is only the chromosomal DNA. So when the conjugation process is occurring, here the plasmid with the chromosomal DNA that is transferred from the donor to the recipient, and in that situation the recipient, it has two sets of chromosome. It's only one set of chromosome that is its own, and another is the donor chromosome that is the recombinant DNA. So sometimes this recombinant DNA may be incorporated within this, or sometimes it need not to be incorporated with this bacterial chromosomal DNA. So here, since the transfer of the bacterial genes is like that, that means two sets of genes that is carried by the plasmid. This plasmid that is transferred from here to this, not this plasmid that is transferred from here to this, it carry two sets of gene. One is the chromosomal DNA of the donor and another is the plasmid factor of the donor. Okay, so that's why this process is called the sexduction. This process is called the sexduction. And F dash plasmid is what? Segments of the bacterial chromosome can become incorporated into the hybrid F plasmids that are called the F dash plasmids. Okay, okay. So here the recombinant, I explained that obviously, that recombinant DNA with the bacterial chromosome and the plasmid, F plasmid factor, this bacteria is called the F dash. That means F dash only that bacteria that has the recombinant DNA. Simple, what is that? So this is the F dash, and here by the conjugation process from F dash another F dash may be processed, may be formed, and in here sometimes this chromosomal DNA may be remain separately from this plasmid, and sometimes this recombinant DNA that is coming from the donor may be incorporated with the chromosomal DNA by the process of recombination. Okay, so conjugation process, the three types of conjugation process I explained in my two videos. So you just check out the first one and obviously this one. Okay, in my next video I will try to describe another topic, and another thing is that if this channel is helpful to you, must share this channel with your friends. And thank you to all of you who have already subscribed my channel and support for the growth of this channel. Okay, so continue with my channel for the next videos. Okay.